With that, we have one more speaker for the session, which is Francois Cinotti from the University of Nottingham. Francois is going to be talking about a Bayesian map of the striatal microcircuitry reveals different wiring processes for D1 and D2 spiny projection neurons. Go ahead, please.
Thank you. First off, I'd like to thank the organizers of this conference. It's great pleasure to be able to present this work, which I've done under the supervision of Mark Humphreys at the University of Nottingham, as just mentioned. So we are interested in a part of the brain which is called the striatum. It's known as the main input nucleus of the basal ganglia. And the basal ganglia is of particular interest because it is supposedly implementing a form of decision making, or at least action selection. The striatum in particular is made up in its great majority of two kinds of spiny projection neurons. So these are neurons which project outside beyond the striatum towards the globus pallidus in particular. And they fall into two different categories. You've got the indirect spiny projection neurons and the direct spiny projection neurons based on their long distance targets. They're also known as D1 and D2 neurons, which is how I will refer to them throughout the rest of the talk, based on the expression of the type of dopamine receptor which they have in their membrane. The classical view of the basal ganglia is that it is a feed-forward circuit in which the cortex stimulates different channels in the striatum. So these different channels correspond to different behaviors or actions. And the role of the striatum is to relay these inputs in two different streams, the indirect and direct streams, towards the globus pallidus. And this means that there is any potential collateral connections between the neurons is completely ignored. So we only assume that the output activity is a reflection of the input activity, and any internal computation within the striatum is ignored. And fortunately for computational scientists who would like to look into the computational properties of the striatum, there are experimental procedures which have been carried out to look at the rates of connections between spiny projection neurons. And here you have a picture from the study by Plenaire 2010, where they labeled fluorescently to either the D1 or the D2 neurons. They then patch different fluorescent or non-fluorescent neurons, stimulate potential presynaptic neuron, and see if they can record a response in the postsynaptic neuron. When they do, they say, okay, these two neurons are connected. They can then report their results in the form of the rates of success of their test. So if you look at the D1 connections in the study by Taverner, you see that in 15% of pairs, they found a connection. And in the case of D1, D2 neurons, it was only about 7%, so half as much. Based on this finding, you have work by Burke and colleagues 2017, who built a simple firing rate model of the striatum, in which they looked at two different subpopulations corresponding to two different behaviors which were being encoded in the striatum, each divided into the direct and indirect pathways. And they assumed an architecture which reflected these discrepancies between direct connections, which are more numerous between direct spiny projection neurons, and between direct spiny projection neurons and indirect spiny projection neurons. And thanks to this architecture, when you have equal cortical inputs, you can find a pattern of behavior which is interesting insofar as even though the inputs are equal, you'll still manage to find a winner. Unfortunately, if the experimenters had taken the trouble of including confidence intervals, you see that the property of connection between D1, D1 and D1, D2 pairs is actually almost certainly the same. And so the question which we want to address is finding a way of correcting this oversight by exploiting as much as possible the experimental data which is given to us to determine the properties of connection. And we're going to adopt a Bayesian inference framework. So it differs from a frequentist inference, insofar as the frequentist inference framework gives us only a single point estimate. So you had the height of these bar plots, which I showed before, which is p hat, hopefully, although it wasn't the case, surrounded by a flat confidence interval. In addition, the single point estimate is usually the maximum likelihood estimate, which is kind of thinking of things backwards, because it takes the data to be the random variable and the p which you're looking for as the variable, which is already fixed. Bayesian inference, on the other hand, instead of giving us a single point estimate, derives a full probability posterior distribution, FP of p, which tells us how likely every possible value of p between 0 and 1 is, given the data and a certain prime. So we're going to start with a prior, which is a beta distribution parameterized with values alpha and beta. You find, for instance, 9 connected pairs out of 34, and you update your beta distribution into a new shape, which has new updated parameters, which corresponds to alpha plus the number of successful connections and beta plus the number of unsuccessful connections. So to illustrate this, we're going to take a prior distribution, which is based on previous literature, and this is one of the key strengths of Bayesian inference. You can use previous knowledge about the system. In this case, we have a study by Kuss and colleagues 2004, which determines that spiny projection neurons connect to one another with a property of 0.12 without distinguishing between D1 and D2 neurons. So we draw a prior which has this shape centered on 0.12, and a certain broadness, which is in this case arbitrary, but we don't want it to be too peak or too broad. And we then plug in the experimental results from the study by Taverner, and we obtain these posterior curves here. So as I said, we now have a property of a description of the probability connection for all values between 0 and 1. And you can see how the blue curves, which represent the posterior curves for D1 neurons, segregate more to the left than the red curves. And so we could say at this point that D1 neurons probably make less connections than D2 neurons. But we can be more quantitative about this by defining a difference between two properties of connection, and the PDF for this difference, for which you have the formula here. So we can start by asking the questions, do D1 neurons actually prefer to connect to other D1 neurons, as was claimed by Taverner and exploited by Perk, by calculating the PDF for this particular difference. And we see that it actually includes 0 in its kind of main domain. And we can also ask the same questions about D2 neurons, and it's the same result, meaning that neither neuron subtype, D1 or D2 neurons, prefer to connect to a particular postsynaptic target. Now that we found there is no postsynaptic targeting, how about we collapse the neuron counts according to the presynaptic neuron? So collapse the 2 red curves and the 2 blue curves and see whether the D1 neurons make less connection to D2 neurons, which is the result which we found here. So here you have the collapsed postsynaptic posterior curves, and here you have the PDF for the difference. And you can see that D1 neurons make connections with the property of about 10%, where D2 neurons have a rate of connection of about 20%. This is a difference which we can track across development, thanks to a recent study by Krzyzewski and colleagues 2019, which looked at rate of connectivity at 3 different stages of postnatal development in the mouse. And you can see here that D1 neurons make their connections very early on. They're already present at postnatal stage 3 to 6, whereas D2 neurons gradually develop their connections over time and eventually overtake the rate of connections of D1 neurons. So if you remember in my first first slide about the experimental procedures, I mentioned a study by Plenner and colleagues, and we could do the same extraction of posteriors for this study. And what we found is that we had posteriors which were smaller than those for Taverner. So this isn't obvious here, but you have to take my word for it. If you compute the PDIs for the differences between the rates of connectivity, you can see they're all slightly offset from 0, except for the light red one here. And so this is an effect which might be due to the different experimental procedures. Taverner limited themselves to a maximum distance of 50 microns, whereas Plenner used a maximum distance of 100 microns. And if you're sampling in a larger area, and if you assume that neurons which are further away are less likely to connect, then this might explain the discrepancy between the 2 studies. And so we wanted to find a way of reconciling the 2 studies. And to do this, we're going to use the following model. It's based on 2 components. First, you have the assumption that the property of connection as a function of distance is an exponential decay function. And secondly, we're going to assume that the experimenters are sampling within their maximum area equiprobably. You combine the 2 and you get this equation here, which relates the property of connection on this axis here, and the beta parameter, which defines how fast the property of connection decreases with distance. So to recap, we have these two studies which have a slight disagreement about the property of connection, which we assume is due to distance. And we want to make sure the 2 studies are in fact in agreement. And to do that, we want to relate the property of connection with the decay rate. So our first ingredient to do this is a way of transforming this property of connection into a decay rate. So we can solve this equation numerically. Secondly, now that we have these posteriors, we want to transform this posterior into posterior for the decay rate. And this can be done mathematically once again, thanks to this equation here. And so if we look at D1 neurons, here you have the posteriors for D1 neurons between the two studies converted into posteriors for decay rates. And you can see that they overlap quite nicely. It's not perfect agreement, they overlap. And here you have the exponential decay curves, which correspond to the peaks of the posteriors here. And you can see they are quite close. And the here you have the half distance shown here, which is quite close in both studies. We can do the same for D2 neurons. The agreement is less evident, but we're at least satisfied the curves are close-ish together. So the estimates lie in the same ballpark. So to conclude my talk, so I ended by saying that there was relative agreement, but it wasn't perfect. So one thing we'd like to test is, instead of using the equiprobable sampling, we're going to use a nearest neighbor distribution. So we're going to suppose that given a choice of neurons within the maximum distance, experimenters are going to prefer to connect neurons which are close together. Psychologically, you'd expect that you're motivated to find connections, so that kind of makes sense. So I've already tested this, and you can reach a better agreement with this method, given parameters which I won't get into that. Secondly, we'd like to complete our map of the striatum by including interneuron data. So we've already done this. There's data from a study by Gittis and colleagues 2010, and there's a very recent paper as well about cholinergic interneurons, which I have to add to this ongoing work. Thirdly, so what we're going towards right now is a map, connectivity map of the striatum, but we'd like to complete it also with a Bayesian analysis of the strength of connections. So for instance, Taverner found that D1 connections are relatively rare, but in addition to this, they're also relatively weaker than other types of connections. So this is something which we could treat with a Bayesian analysis using, I suppose, a Gaussian prior. So this is not done at all. This is really out there. And lastly, if given time, all of this data could then be used to draw a new model of the striatum in which we take into account the lateral connections and their potential effects on the computations which take place in that part of the brain. Thank you very much for your attention.
Thank you very much, Francois. Everyone, please feel free to ask any questions. You can put your virtual hand up or use the Q&A function in the Zoom. And that was some perfect timekeeping, by the way. I didn't even have to put my hand up. Again, panelists, feel free to also ask questions if you do have any. Okay, I don't see anything coming up at the moment. I'm not sure if people are typing very long questions or if your talk was just very, very clear, Francois. All right, I think with that we're done then. Do feel free, obviously.